Okay, hello everyone. In first video of transpiration unit, we have discussed regarding introduction of transpiration, definition as well as types of transpiration. In this video, we are going to discuss regarding structure of stomata and mechanism of opening and closing of stomata. So let's start with the structure of stomata. So look at the slide. There is a structure of stomata or open stomata as well as closed stomata. So let's discuss regarding actual what is there in stomata. Stomata are the minute pore or opening which are present in the epidermis of leaf. Each stomata consists of elliptical or we can say crescent shaped pore surrounded by two specialized cells called as guard cells. So here you can see these two this one and this one. These two cells are the specialized cells called as a guard cells. Each guard cell is kidney shaped as in dicot and डम्बेल शेप्ड इन मोनोकॉट. डायकॉट प्लांट मध्ये तुम्हाला किडनी शेप्ड किंवा बीन शेप्ड गार्ड सेल्स बघायला मिळतात, व्हायल इन केस ऑफ मोनोकॉट प्लांट्स डम्बेल शेप तुम्हाला स्टोमाटास बघायला मिळतात. सो ईच गार्ड सेल्स कंटेन्स न्यूक्लियस, क्लोरोप्लास्ट and lining layer of cytoplasm and central vacuole filled with cell sap. Look at here. Look at the diagram. Look at the diagram. Here there is this is a guard cell. This is a guard cell. This guard cell consists of, this is vacuole, this is chloroplast, and this is nucleus. So these guard cells are specialized cells which are responsible for formation of a spore or a opening. Each guard cell contain a large amount of protoplasm than their neighboring epidermal cells. So neighboring epidermal cells, these are the neighboring epidermal cells. We can call them as a subsidiary cells. So the inner wall of guard cell is thicker and non-elastic. Look at the inner wall. This is the inner wall of guard cells. Just a minute. This is the inner wall of guard cells. This one and this one is the inner wall of guard cells, which is quite thick in size and which is non-elastic, while this is the outer wall of guard cell which is thin and elastic. So the structure of inner wall and or nature of inner wall and outer wall is very much essential. As the outer wall is thin and elastic, during opening of stomata it plays an important role. So you must know this character of stomata. Okay. So this is all about structure of stomata. Now next point from this topic is opening and closing of stomata. We are going to refer these diagrams for opening and closing of stomata also. So let's see the next point, opening and closing of stomata. So the opening and closing of stomata is also known as a stomatal movement. The inner wall of each guard cell is thick and non-elastic, as we have discussed in earlier slide, while the outer wall is thin and elastic and can be stretched. So guard cells always have stretching starch containing chloroplast, but surrounding epidermal cells do not contain starch containing chloroplast. For example, look at this diagram where these are the guard cells are having a chloroplast with starch, while these epidermal cells do not have a chloroplast with starch. So starch plays an important role in opening and closing of stomata. Let's see how. See, during the daytime when there is a light, starch in the guard cell is converted into sugar. Look at this diagram, flow chart. Okay. For example, glucose. This conversion takes place in presence of inorganic phosphate, and during this an enzyme called phosphorylase acts as a catalyst. That means starch plus PO4 is converted into glucose in presence of light and phosphorylase. So this reaction or this conversion of starch and PO4 into glucose leads to opening of stomata by increasing a turgor pressure or osmotic pressure of a guard cell. Glucose being soluble in water increases the atmospheric pressure of the guard cell. As a result of this, the guard cells draw or take up water from the neighboring mesophyll cells. Due to diffusion of water into guard cells, the guard cells become turgid. The outer wall of guard cells, being thin and elastic, gets stretched or pulled outwards, while the inner non-elastic wall, thick wall of guard cells becomes concave. As a result, the two guard cells move apart from each other and the stomata opens widely during daytime. When the stomata are open, the water in the vapor form from intercellular cells is lost to the outer atmosphere. So, look at this diagram where stomata is open. स्टोमाटा कधी ओपन होतो, जेव्हा स्टार्चचं कन्वर्जन इथे गार्ड सेल्समध्ये स्टार्चचं कन्वर्जन ग्लुकोजमध्ये होतं, तेव्हा हा जो काही ग्लुकोज आहे किंवा शुगर फॉर्म आहे increases the osmotic pressure by absorbing water from these epidermal cells. ठीक आहे, शेजारच्या एपिडर्मल सेल्समधलं वॉटर जे आहे तर ते गार्ड सेलमध्ये डिफ्यूज होतं आणि गार्ड सेलमधला जो काही शुगर आहे ते वॉटरमध्ये सोल्युबल होतं आणि जी काही गार्ड सेल आहे तर ती थोडीशी स्वेल होते, फुगते आणि ह्याच्यामध्ये जी काही इनर वॉल आहे ती नॉन इलास्टिक आहे, ज्याच्यामुळे इनर वॉल रिमेन्स ॲज इट इज व्हाईल द आउटर वॉल जी थिन आणि इलॅस्टिक आहे ती स्ट्रेच होते, ज्याच्यामुळे ह्या ज्या दोन्ही गार्ड सेल्स आहेत किडनी शेप त्या एकमेकांपासून अपोजिट डिरेक्शनला खेचल्या जातात आणि मध्ये एक पोअर किंवा होल ओपन होतं आणि दॅट इज द ओपनिंग ऑफ स्टोमाटा. So let's see, and this opening of stomata happens during daytime. Now what happens during night time? During night time, water from guard cells enters the subsidiary cells and as a result guard cell become flaccid due to decrease in turgor pressure and the stomata get closed. Let's see it quite thoroughly. Look at the slide here. In dark condition, just a minute. During night time, sugar of guard cell is converted into starch in presence of enzyme phosphorylase. जसं डे टाइम मध्ये स्टार्चचं कन्वर्जन ग्लुकोजमध्ये होतं, एक्झॅक्टली अपोजिट डार्क कंडिशनमध्ये ग्लुकोजचं कन्वर्जन स्टार्चमध्ये केलं जातं इन द ॲब्सेन्स ऑफ लाईट, दॅट इज इन डार्क कंडिशन विथ द हेल्प ऑफ एन्झाईम फॉस्फोरायलेज. सो स्टार्च इज इनसोल्युबल अँड ऑस्मोटिकली इनॅक्टिव्ह सबस्टन्स, देअरफॉर ओपी ऑफ गार्ड सेल इज डिक्रीज्ड. स्टार्च वॉटर ॲब्सॉर्ब करत नाही, त्यामुळे गार्ड सेल्सचं जे काही ऑस्मोटिक प्रेशर आहे किंवा गार्ड सेल स्वेल होत नाहीत, त्यामुळे आउटर वॉल स्ट्रेच होत नाही, इट रिमेन्स ॲज इट इज फ्लॅसिड. सो बोथ द गार्ड सेल्स रिमेन्स फ्लॅसिड, त्या चपट्या होतात, ज्याच्यामुळे तिथे स्ट्रेचिंग ॲबसेंट असल्यामुळे स्टोमाटा जो आहे तो पोअर जो आहे तो बंद राहतो. सो ॲज अ रिझल्ट वॉटर डिफ्युजेस फ्रॉम गार्ड सेल इनटू नेबरिंग मेसोफिल सेल्स. ॲज स्टार्च इज प्रेझेंट इन द किडनी शेप्ड गार्ड सेल्स, because it does not absorb or it get solubilized into the water is loose or diffused to the near epidermal cells. सबसिडरी सेल्समध्ये वॉटर बाहेर टाकलं जातं किंवा डिफ्यूज होतं, ज्याच्यामुळे गार्ड सेल्समध्ये कुठल्याही प्रकारचं स्वेलिंग किंवा ऑस्मोटिक प्रेशर इन्क्रीज होत नाही. अँड सो the pressure developed in the cells due to osmotic diffusion of water inside the cell is called as a turgor pressure. As a result, the guard cell becomes flaccid. Due to flaccidity of guard cells, stomata is partially or completely closed during night. And when stomata are closed, the water in the form of vapor from intercellular space is not lost through stomata to the outer atmosphere. So, sugar to starch and starch to sugar conversion are independent on pH and temperature of guard cells. So, during daytime, the pH of guard cells remains 7, that is neutral or alkaline. It favors the conversion of starch into sugar. So, when sugar is present in guard cells, stomata are open, but at night time, CO2 is released in the guard cells during respiration, and CO2 is not used for photosynthesis during night time. So, due to release of CO2 in guard cells, the pH of guard cells becomes acidic, that is up to 5, and the acidity of guard cells favors conversion of sugar into starch at night time. So, when starch is present in guard cells, the stomata are closed, and loss of water is prevented through stomata at night. So, thus the opening and closing of stomata depends upon the change in turgidity pressure of guard cells. If there is an increase in turgor pressure of guard cells, stomata will remain open, and if turgor pressure decreases when starch is present in the kidney shaped guard cells, at that time stomata will remain closed down. It is known as a sugar starch hypothesis of opening and closing of stomata. Okay, now look at the slide. I have given the flow chart, what happens during night and what happens during light. In light condition, photosynthesis is going on, so CO2 is consumed. So as CO2 is consumed, there is a decrease in CO2 concentration, increase in pH up to 7. So in this pH, the starch is converted into sugar and increases the cell sap concentration, as due to which endoosmosis happens, means water comes from nearby epidermal cells into the guard cells, and the turgor pressure of guard cells increases. As turgor pressure increases, it exerts pressure on outer thin elastic walls of guard cells, and stomata get kidney shaped guard cells get stretched, and so that stomata get open. While in case of dark condition, no photosynthesis there is an increase of CO2 concentration, decrease in pH. So decrease in pH leads to conversion of sugar to the starch, and this starch does not absorb water, or it is water insoluble, so that the cell sap concentration decreases, and as the decreased cell sap concentration is there is exosmosis, means water is diffused from guard cells to other epidermal cells. Water goes out of the guard cells. That's why turgor pressure decreases, and when turgor pressure decreases, the guard cells become flaccid, flat, ज्याला आपण चपटे असं म्हणू शकतो, ज्याच्यामुळे ड्यू टू विच द स्टोमाटा गेट क्लोज्ड. ओके, so this is in short the mechanism of opening and closing of stomata, or we can say movement of stomata, and this mechanism, which is based on sugar and starch conversion, that's why it is known as a sugar starch hypothesis of opening and closing of stomata. There are two more hypotheses regarding stomatal opening and closing or stomatal movement that we are going to discuss in the next video. So we will stop here for today. Thank you.